Hey everybody, thank you for joining me for another lecture video. We're going to be discussing the lymphatic system and immunology today. So looking at our lymphatic system and the immunological process. So basically what we're talking about here in immunology is host defense, or the ability to combat disease. So for our immunological concepts, we look at the types of cells that are there and the types of organs that are going to assist with their function. So what we're going to be doing here, right? Cells and organs. We have the immune system. It is going to contain primary lymphoid organs, such as the spleen and the thymus, secondary lymphatic tissues, and various cells of the innate and adaptive immune systems. Our key primary lymphoid organs, as mentioned, we have the thymus. This is the site of T cell maturation. We have the bone marrow, the site of white blood cell production. Secondary lymphatic tissues, examples of this, going to be things like the tonsils, the lymph nodes, the lymph vessels, and the skin. All right, so we can go ahead and take a look at our immunology tutorial here. Hopefully it'll play.
Let's explore your future with Udemy.
Yeah.
Hi there, welcome to a short overview on the differences between the innate and adaptive immune systems. This is the very first video of my Immunology series. In fact, the very first video of this channel, Breathing Bio. Let's have a look. So let's imagine the immune system to be the army of the human body or host. Think of innate immunity as the forgetful soldier, no memory, merely rushing off to war and fighting left, right, and center, acting rapidly in every sense of the word. No specific target. In fact, this section goes at anything not belonging there. In other words, foreign invaders. Your adaptive immunity, on the other hand, is far more specialized. In fact, almost like an intelligence unit of the military. Their fighting skills are far more specific, far more targeted at specific foreign invaders. They take time to respond as they are far more calculated, planning every step as they go.
All right, so you see now that we have the two portions of the immune response, innate and adaptive immunity. So here, for example, are some of the cells that we see, all right? We have the classical immunological pathway and the alternative pathway. These are going to be adaptive immune cells. So here we have CD4 T cells, right? These are going to be down the classical pathway of antibody production. So talking about the specific cells and organs, again, we start with the primary lymphatic tissue of the thymus. The thymus is going to be the area where the T cells become educated, so to speak. This is going to provide an environment for those T cells to mature. Thymus is going to be the most active and largest during the neonatal period and also early childhood. After that, the thymus is going to begin to atrophy, and the actual body of the tissue is going to be replaced by a lot of fatty tissue and adipose that comes in and fills the space. However, even though the thymus diminishes in size and in population of thymus cells, we still have the potential for T lymphopoiesis, which is creation of mature lymphocytes. The bone marrow is our secondary tissue. This is going to be a tissue that is found inside of the cavities of bones. We're going to produce our formed elements of the blood here. So red blood cells in the red bone marrow. The red bone marrow is also a key element of the lymphatic system. This is one of the primary places that we generate lymphocytes from immature hematopoietic progenitor cells. So the bone marrow and the thymus are going to constitute the two places where we are going to be able to mature lymphocytes. The B lymphocytes will mature in the bone marrow. The T lymphocytes migrate to the thymus and mature there. Our lymphatic system as a whole is going to be a circulatory system, okay? It is going to circulate what we term lymph fluid. The lymph fluid is going to flow around the lymphatic vessels, and it is going to work very similarly to how our cardiovascular system works with carrying the blood. The lymph fluid is going to be a filtrate from the plasma of the blood. So we're going to get the plasma to filter out into the interstitial space between tissues, and that filtration is then going to be picked up by lymph vessels. We have several different functions of the lymphatic system. Again, filtering the fluid out from the interstitial space. It's going to be a large area of white blood cell concentration. So we can do a lot of immune detection, pathogen destruction here. We're going to transport those white blood cells around. We transport the antigen presenting cells such as dendritic cells to the lymph nodes. We can find lymphatic tissue in almost any organ. It's going to have some sort of resident immune cell, whether it be a macrophage or a neutrophil. But our main places of lymphoid tissue are going to be in the primary lymphoid tract, which is going to have lymph nodes along the vessels. Next is going to be the spleen. The spleen is also a major activator in terms of immune function. We see the filtration of red blood cells and white blood cells here. It is going to be similar in structure to the lymph nodes, and it's going to act as a primary filter for red blood cells. So it takes the old and dying red blood cells out of the blood and gives them a place to go and destroys them. It is also going to be a place where we can synthesize antibodies. It has a red pulp that is going to be the destruction of the red blood cells, and it has a white pulp. And in the white pulp, we have antibody synthesis, right? It's also going to be able to remove our antibody-coated bacteria with the antibody-coated blood cells. Next tissue type is going to be the tonsils. We have several different tonsil populations. We have the palatine tonsils. We have the nasopharyngeal tonsils. All right, and we also have the lingual tonsils. Palatine tonsils are going to be at the back of the throat on either side. The nasopharyngeal tonsil is going to be at the back of the nasal cavity. This is also known as the adenoid tonsil. The lingual tonsil is going to be at the base of the tongue. Tonsils are going to be what are known as immunocompetent tissues. So they're going to be tissues that are able to ingest and degrade pathogens if they come across it. They're going to be open to the cavities of the body that are going to intake from the outside environment. So we have a first line of defense here as far as if we are taking in foreign particles. Next is going to be the lymph nodes. Lymph nodes are widely distributed across the lymph system. They're going to be at a lot of different areas. They're going to be maintained in large populations in certain regions, however. Places such as the axillary region of the armpit, the cervical region of the neck, the inguinal region of the groin. Those are going to be areas with large lymph node populations. The lymph nodes are going to contain massive quantities of immune cells. So they're going to be a place where populations of B cells, T cells, and other immunological functioning cells are going to be present and able to combat the pathogens. Right? Lymph nodes are going to act as the filter for the lymph circulation. So the lymph fluid is going to arrive at the lymph nodes via the lymph vessels, and here we are going to filter out that lymph and try and find any sort of foreign debris or particle or pathogen that could potentially be harmful. In the lymph node, we have a very large proportion of macrophages, which are phagocytic cells, meaning that they're going to take in and ingest any foreign particles that they find. They are large cellular eaters. They will engulf any foreign particle, take it in, and try to degrade it. We can't go on without failing to mention the skin, of course, as it is the initial line of defense against anything from the outside environment. It's going to be able to combat the outside environment via the Langerhans cells or dendritic cells within the epidermis. Those are going to be the part of the adaptive immune response. Next is the liver. The liver, as it is with most systems, has a wide variety of functions. In immunological sensation, it is going to be the reticuloendothelial system. This is where we are going to have immunologically active cells. So this is a place where we can send antigens, and we are going to store those antigens and carry them via the portal system of the liver. All right. So talking about the cells of the immune system, we've already mentioned the lymphocytes, and we did talk about the macrophages briefly. But when we're talking about white blood cells as a whole, we're talking about leukocytes. So a leukocyte is a white blood cell. These are going to defend against the body, or sorry, defend against the pathogens that are trying to infest the body. We have 5 different types of leukocytes. We've already mentioned a couple of them. We'll go on to mention a couple more. These are all produced in the bone marrow. The innate leukocytes are going to be leukocytes of the innate immune response or the innate immune system. These are going to include the neutrophils, the macrophages, the mast cells, eosinophils, basophils, and natural killer cells. All right. These are going to be the cells that mount the first response and are able to attack pathogens as soon as they enter the body. The neutrophils and macrophages are the phagocytic cells. They are also going to be the first responders, so they're going to come across whatever is there first, and they're going to try and deal with it on their own. The neutrophils are going to be found in high populations in the bloodstream. They are usually about 50% of the entire white blood cell population, so they're going to be very abundant. During the acute phase of inflammation and infection, the neutrophils are going to get there first, and they're usually going to be able to ingest and degrade a pathogen and try to present that antigen on the surface. Macrophages are going to be a little bit more regional. They usually migrate and reside within tissues and usually stay there. So they're usually resident in a certain tissue type. Some of them, however, are going to be acting as scavengers, meaning that they will infiltrate into areas that they are needed and they will migrate there if they are signaled. They will go around ingesting foreign debris via phagocytosis, eating it, and trying to present that antigen to other cells. Dendritic cells are also phagocytes. We mainly find these in the areas exposed to the external environment, so the skin, the nose, the lungs, the stomach, and the intestines. These are going to be a link between the body tissues and the innate immune response. So they are able to present antigens to T cells. So they are part of the adaptive immune response if they are able to take in a pathogen and are able to present that pathogen on the surface of the cell. Next is the mast cell. Mast cells are going to reside in a lot of connective tissue areas and mucous membranes. They're going to help to regulate the inflammatory response. So they're going to secrete histamines and heparins, which are able to increase inflammation and also dilate blood vessels. They're most often associated with allergic and anaphylaxis responses. So when we have an allergic response, usually we take an antihistamine against histamine. Histamine is a cytokine that is released by the mast cell. So this is what we are targeting when we take that. Basophils and eosinophils are going to be granulocytes. The neutrophil, basophil, and eosinophil are all 3 granulocyte cells. Basophils and eosinophils are going to be related to the neutrophil. They are going to be involved heavily in things like parasitic responses and allergic reactions. Natural killer cells are going to be leukocytes that differentiate after they find a tissue that has been infected by a virus. So they're going to be very active in viral infection as well as in cancerous growth. The adaptive immune response is going to also be our leukocyte populations of lymphocytes. As we have mentioned previously, our B and T cells are the 2 types of lymphocytes, and they're also going to be produced in the hematopoietic stem cells in the bone marrow. How T cells work is that they will recognize non-self, quote unquote, cells from self cells or self-presenting cells. So a non-self cell, for example, is a bacteria or a virus that is in there that should not be in there, all right? And only after the antigens have been processed and presented in combination with a quote-unquote self-receptor, are we able to target them? So the T cell is going to find pathogen. It's going to find one that has been ingested and presented on the surface. And when it is presented on the surface, it is presented in what is called an MHC, or major histocompatibility complex. And this is going to be how the antigen of the pathogen is going to be recognized. We have two subtypes of T cells. We have what is known as the killer T cell. This is going to come along and destroy cells via cytotoxic chemicals. And then we also have the helper T cell. Helper T cells are going to regulate the innate and adaptive immune responses and go a long way towards activating B cell antibody production. The T cells do not have any cytotoxic activity, so they are not able to actually go on and kill infected cells or pathogens directly. A third subtype is going to be the gamma T cells, and those are going to recognize intact pathogens that are not bound to MHC receptors. So these are going to be not very numerous or populous in concentration. Next are B cells. B cells are going to only be activated when they find a specific antigen that they recognize. So they have to be able to produce an antibody that is specific for an antigen that's been recognized. If there is no antibody, then there is no B cell activity. So the antibody is going to have to recognize a pathogen without any need for the antigen to be processed. Each time that we have a lineage of B cell, it's going to express a different antibody. So the complete set of B cell antigen receptors are going to be able to present all the antibodies that the body can potentially manufacture. All right. So when we talk about the immune system as a whole, it's going to be the system of biological processes and structures that are able to protect against disease. Okay. We protect against a lot of variety of things like viruses, bacteria, parasitic worms, and we have to be able to distinguish them from our own tissues. So we have multiple defense mechanisms that we're able to use, right? We have to be able to get an initial response from the innate immunity and then an adaptive response over time from the T cells and B cells. All right. Other immune mechanisms that we have antimicrobial peptides that are known as defensins. These are going to be a large component of immunotherapy. Whenever we have an immune system issue or we have a pathogen that we have little ability to combat, defensins are able to go in and attack a pathogen without recognizing it, which means that pathogens and microbes don't really have the ability to change their setup or change their conformation so that that defensin does not recognize it, because the defensin doesn't necessarily care what the pathogen looks like, it's just going to attack it. So defensins are going to be able to attract and kill pathogens at a pretty decent clip. We also have the complement system, which is going to be a bunch of proteins that are going to be assisting in the innate immune response. Our adaptive immunity is termed adaptive because it is acquired, because it has to take place over time. We have to be able to find a specific pathogen and create an antibody response to that pathogen's antigen. We then have to have some sort of a memory. So we produce B memory cells, T memory cells that are able to take that response and then kick it on again if we come into contact with the pathogen a second time. So for our adaptive immune response, we have to be able to protect against an organism if it comes back again. So we have that memory that comes into to play with the B and T memory cells. All right. The innate immune system is going to protect against responses from our physical barriers and from our cell-mediated and humoral immune responses. So as our chart shows, if we have contact with some sort of an infector, right, Initially, we are going to have a massive immune response from the innate immune system. Okay. Over time, we are going to be able to produce antibodies and effector T cells. If we come into contact with it again, right? Notice how at this point, our immune response is much faster. So instead of it taking several days for this antibody and effector T cell population to start, we have within a few hours to a day, that immune response is going to kick on again. Then if we have it a couple of years later, if we have formed a strong enough immune response to it, then it will only take a short amount of time for those antibody and T effector cells to be activated once more. So you can see that subsequent infection is going to lead to a much faster and stronger and a more effective immune response than our first exposure was. So if we have a successful invasion, the adaptive immune response is going to be key in trying to eliminate that. Both our innate and adaptive immune responses depend on the ability to distinguish between self and non-self molecules. This is where we have autoimmune disorders that come into play. The autoimmune disorders are going to be because our body is not able to recognize what is self and what is not. And we start to attack our own cells or our self cells because we have no way to distinguish between what that tissue is and what we are not supposed to be attacking. So our immunity involves both specific and non-specific components. If we are going to go with our two categories of innate and adaptive immunity, when we get adaptive immune responses, there are going to be a lot of subsections to this. They're going to occur in two ways. We can either do it naturally, we come across the pathogen or whatever in our natural environment, in our everyday lives, or we can do it artificially, as in medically induced. So naturally, we can get it in two ways. We can do it active infection, where we live about our normal lives and then come across it in nature, or we get it passively if it's passed from mother to fetus during pregnancy. For artificial, it's going to be either passive or active. Active is going to be something like a vaccine or immunization, where we artificially inject the pathogen into our bodies and it's able to mount an immune response. Or it's passive, where we get the antibodies that are already made from another source, transplant them into our bodies, and then we have an immune response that we have supplemented into our bodies for a particular pathogen. So if we think about that, the passive transfer of antibodies is only going to be a temporary fix because we don't have the ability to produce those antibodies for our own self cells again. So as soon as that cell population is used up, now we can't mount a response again. Same thing for passive natural immunity. If we have it passed down from the mother to the fetus, once those are used up in the fetus and the baby, they're not able to be produced again because the baby's cells are not the ones that produced it, so they can't reproduce the antibody later on. So our innate immunity is going to be the natural resistance we have. Whenever we have a microbe that's being first encountered by the epithelial layers, we are going to initiate that response, right? We have the traditional inflammation response from chemical signals known as cytokines. This is where we have the fever, swelling, redness, whatever else that's going to happen. Phagocytic cells are the first responders for that. Again, our neutrophils and macrophages are going to come about, try to ingest the invading microbes and get rid of them. All right. So adaptive immunity, again, we have two major subtypes to that. We have the cell-mediated response, which is the T cell response, and we have the humoral immunity, or the B cell response, right? So our B cell response is going to be kicked on by the helper T cells once we are able to identify that antigen. All right. So both naturally and artificially acquired immunity are going to be able to be subdivided. The passive immunity is going to be acquired through a transfer. This is usually only going to last a few months, as I stated, because we do not have the ability to create those cells on our own. Active immunity is going to last much longer because we have our own host cells that are able to identify the antigen and then produce a memory for that. So this lasts a lot longer, sometimes for an entire lifetime. All right. So we characterize our adaptive immune response into humoral immunity, which is the B cell response by antibodies, and cell-mediated immunity involving the T lymphocytes. Humoral immunity is going to be activated when we are generating our own antibodies. The cell-mediated immunity is activated when the organism's own T cells are stimulated. All right. Our last cell type that we can discuss is the natural killer cells. These are part of the innate immune response. They're going to recognize MHC1 molecules on infected cells and tumor cells. We have the lymphocytes, all right. Natural killer cell is going to be a subdivision of the lymphocyte or white blood cell. The infected cells, if we have, let's say, a virus enter the body, are going to be targeted and destroyed immediately by the natural killer cells if they see an abnormal MHC1 molecule on the cell surface. So, natural killer cells are very good at finding viral infected cells and also tumor cells abnormally dividing, because they will have a cell surface antigen that is going to be a little bit wonky or a little bit abnormal. And if they're able to see that antigen that's weird, or the MHC molecule that's strange, they will immediately try and destroy that place. So while they are part of the innate immune response, they're best understood because of their relatives, which are the T cells, right? The T cells are going to be very close to natural killer cells in function, and they are also close to them in structure. T cells specifically are going to be the lymphocytes that mature in the thymus. They're able to identify intracellular infections. They're very good at identifying viral infections, again, because of that altered MHC complex on the surface of the infected cells. The MHC molecules are proteins that are present on the surface of the cell membrane, and they're going to help us identify self versus non-self. So if we have an infected cell, that MHC1 molecule is going to display fragments of proteins from the infectious agents. The healthy cells don't display any of those proteins, so they're ignored. But if we do have those protein fragments in the MHC, then the T cell is going to find it, try to destroy it, and attack it. All right, an infected cell or a tumor cell is usually incapable of synthesizing and displaying MHC1 molecules. So, if we do not have MHC1 on the host cell surface, then we are going to identify that with our T cells, and we are able to attract them, right? If we deplete the MHC1 molecules, we will prevent the T cells from recognizing them. So if viruses are able to infect a cell and are able to destroy or get rid of all of the MHC complexes on the cell surface, then the T cells don't actually have the ability to recognize them anymore. So they will overlook them because there is no MHC molecule left anymore on the cell surface for them to recognize and destroy. However, if there are no MHC molecules on the surface, the natural killer cells are able to see it, okay? So if they find a cell or they come across a cell that has no MHC molecule at all, then they will dictate that this is an abnormal cell also, and they will be able to destroy it, where a T cell was not able to recognize it and destroy it. So natural killer cells offer a complementary check for our unhealthy cells, right? If the T cells don't have an MHC complex to identify and resolve, such as in the case with our viral infection, then they will not be activated. But the natural killer cells are able to come along and sort of fail-safe or proof that by seeing a cell that doesn't have an MHC complex at all and recognizing that that is abnormal and that should not be the case. So also with tumors, with cancerous formations, we have dramatically altered genetic expression for those tumor cells. That will lead to a deformed MHC complex. Natural killer cells are then able to target that tumor cell or that tumor growth and are able to try and destroy it. Right? Natural killer cells are always going to be active. They're going to be present in the blood in most cases. Whenever they come across a normal MHC complex or a self-MHC complex, if they do happen to come across it and they happen to attach to it, this self-intact MHC molecule will be able to negate the natural killer cell from releasing its cytotoxic chemicals. And that will cause the natural killer cell to move on to another cell population because they realize that this cell is healthy and should be there. After we have a natural killer cell detecting an infected or tumor cell, it is able to secrete granules from the cytoplasm. These granules are comprised of a protein known as porphyrin, and this is going to be able to destroy the cell membrane of the target cell. We also have granzymes released. Granzymes are going to be released in the immunological synapse. A granzyme is a protease. This is an enzyme that degrades proteins. It is able to digest the cellular proteins and will digest the cellular proteins of the target molecule, which will in turn cause it to undergo apoptosis or programmed cell death. Natural killer cells are constantly going around the body, and if they do find a cell type that is not recognizable or a cell that is abnormal, they will secrete these granules and they will try to destroy it. All right. They are very effective for controlling potential infections and preventing any sort of cancer progression. All right. That was the basis of the immune system and of the lymphatic system as well. Thank you all for joining me, and I appreciate it. Like and subscribe to the channel. Go to the Patreon, patreon.com/twistedscience for more. And also thank you for your time. I'll see you next one.